Good morning, everyone. Today, we will talk about a very important part of our body's defense system, the membrane attack complex, or MAC. MAC is a vital tool of our natural defense system. It is the final product of the complement system. The MAC creates cytotoxic pores on the surface of microbes to eliminate them. Our immune system uses the complement system. This is part of our innate immunity, which is a mechanism that quickly identifies and removes pathogens and damaged host cells. The complement system has more than 35 proteins circulating in your blood or bound to cell membranes. The most important proteins are named C1 through C9. This system has an important influence on host defense and inflammation. There are three major pathways of complement system activation: the classical pathway, the lectin pathway, and the alternative pathway. These three pathways can all activate the terminal pathway. The MSC is formed during this terminal pathway. MSC consists of five complement proteins: C5b, C6, C7, C8, and C9. C5b is the initiator. It acts like the foundation or the starting point for the complex. C6 and C7 bind to C5b and extend the structure. C8 inserts into the membrane and anchors the MSC to the cell membrane. C9 is the primary effector. Many copies of C9 form the transmembrane pore. This is the whole process of assembly of MSC. The assembly begins with the cleavage of C5 into C5a and C5b by C5 convertase. Then C6 binds the C5b intermediate and forms the C5b6 complex. After that, C7 binds the C5b6 complex and the newly formed C5b7 complex, which is anchored to the membrane surface. Subsequently, C8 is added to the complex to form C5b8. In the last step, many copies of C9 take part in the assembly, and these C9 molecules traverse the membrane to form the final MSC. The MSC forms transmembrane pores, permitting water and ions, particularly sodium and calcium, to enter the cell. This quickly causes the osmotic pressure inside the cell to become very high. This pressure induces cellular swelling and rupture. This entire process is known as osmotic lysis. The MSC is particularly effective against gram-negative bacteria. Gram-negative bacteria have a defensive wall, the LPS layer, but the MSC pores bypass the LPS layer and directly attack the cell membrane. The MSC is important in causing different diseases. MSC is essential for eliminating pathogens, but if the activation is excessive, it can lead to tissue damage. And then, this can cause problems like autoimmune, inflammatory, and neurological diseases. Since MSC is so powerful, our own cells must be protected. One key protector is a protein called CD59, also known as protectin. It is found on the surface of our own cells. CD59 binds to C8 and C9 and inhibits the polymerization of C9. This prevents the complete formation of the pore on our own healthy cells. In summary, the MSC is the final effector of the complement system. It is built sequentially on the membrane using C5b through C9. Its function relies on osmotic lysis to destroy pathogens. The MSC is crucial for clearing invaders and keeping us healthy. Thank you.